Hello, this is Rich Eisenberg. I'm editor-in-chief of Inorganic Chemistry. As part of our 50th anniversary year, we're bringing to you interviews with leaders in our field. Today, it's a distinct pleasure to have as our interviewee Professor Joan Valentine from UCLA. Joan is the editor-in-chief of Accounts of Chemical Research and is also a member of the National Academy of Sciences and the American Academy of Arts and Sciences. Joan was a winner of the Alfred Bader Award for Bioinorganic Chemistry and also a medalist for the John C. Baylor Medal from the University of Illinois. Welcome, Joan. It's a pleasure to have you here.
Thank you. It's a pleasure to be here. Good.
Joan, let's tell our viewers how you started, the early years before you became professionally involved in chemistry, your education as a child, how you developed your interest in science.
Well, actually, I wasn't all that interested in science. You know, Sputnik happened. I'm one of those Sputnik children, but I mean, a little bit too old to have been inspired. But so the science wasn't emphasized in school. But both of my parents were physicians, and they, so I was going to be a doctor. I was going to be a medical doctor. And so that was my goal, and I was quite set on it. Actually, all the way until I was a junior year undergraduate in college. I was interested in the little bit of science I had, and I had a chemistry set, but it never occurred to me that I would do chemistry. But then when I was in college, I was taking analytical chemistry from George Fleck, a professor at Smith College, and I finished all the analytical chemistry early, and so he said, oh, well, I'll give you a research project. This was also before undergraduate research, right? So we weren't routinely doing undergraduate research. And so he gave me a problem, and I said, you mean nobody knows the answer to this? And he said, that's right. And that changed my life. I mean, I can solve problems where people don't know the answers. And, you know, it was inorganic chemistry, and that's what started it. And, of course, then there are the colors and so on. I was hooked.
But it's the role of a teacher who asked a question. And so tell us a little bit about the graduate work that you did and what you studied and where you did it.
So I got as I said, I got my undergraduate degree at Smith College, and then I went to Princeton University for my PhD in inorganic chemistry. And there, so my entire career I've worked on metals and O2. It all started when I was actually a graduate student, when I was doing inorganic photochemistry on cobalt dioxygen complexes. And I got very interested in how metals bind O2. And during the period that I was at Princeton, I actually spent some time at Stanford also in Jim Coleman's lab and worked on some other metal O2 complexes. And I've worked on metal O2 complexes and related things my entire career, actually, since then.
So those are actually some of the important threads and themes that will run through all of your research.
Yes, that's true.
Now, I know that one part of it relates to superoxide and superoxide dismutase. How did you pick that particular problem and focus on it?
Okay, well, Tom Spira doesn't remember this story, but actually I was, but it's true. I was an starting assistant professor at Rutgers University, and I had been working on metal O2 complexes, but when I started, there weren't very many, but by the time I was starting as an assistant professor, I mean, when I started as a graduate student, everybody was doing this. And I was talking on the phone to Tom Spiro, and I said, Tom, if I work on metal O2 complexes as a assistant professor, everybody's doing that. And he said, you know, I just got back from a really interesting Gordon Research Conference, and this guy called Erwin Friedovich talked about an enzyme called superoxide dismutase. And I had never heard of it before. It was, it was really, literally, had just been discovered by Friedovich. And so then I thought about, O2 minus. I'd already worked on O2 minus complexes of cobalt. I had no idea it was biologically relevant, but that's what started me.
Now, the superoxide dismutase relates in some ways to ALS. You've been working on that. Can you explain to our viewers first about ALS and how the superoxide dismutase work ties into that?
Okay, so actually in the beginning of my academic career, I was doing inorganic, I was trained as an inorganic chemist, not a biochemist. And I was doing inorganic chemistry, metals and superoxide, and hoping that the chemistry would be biologically relevant. But then I had a graduate student who signed up to work with me at Rutgers, and named Mike Pantoliano, and he was a biochemistry graduate student. And I told him, I don't know any biochemistry. And he said, it doesn't matter, I want to work for you anyway. And I said, okay, but we're not going to work on biochemistry. And he said, okay. And then he proceeded to isolate copper zinc superoxide dismutase. And he said it was just for fun, he wouldn't work on it. But then he showed it to me, and there it was, and it was blue-green. And I said, oh, that's just a molecule. It's a coordination complex, and we started working with it then. So then we worked for many years on copper zinc superoxide dismutase. The bovine, the isolated from cows, and when I moved to UCLA in 1980, I was looking for a way to expand. We actually, the technology had been developed to site-directed mutagenesis, and so that we could actually make mutant forms of copper zinc superoxide dismutase. So then we were doing protein redesign, and again, inorganic coordination chemistry was what it was about. But we, right in 1993, there was the announcement that mutations in the gene coding for copper zinc superoxide dismutase caused ALS. ALS is Lou Gehrig's disease. I knew very little about it at the time. But this was extraordinary. Nobody had ever linked these, but we were in a very good position because we were already doing mutagenesis. So we could actually make the mutant proteins and study them, and that's what transitioned us into the ALS field.
I'm going to go back a little bit and ask you to explain a little bit of why superoxide dismutase is important in terms of our being able to live.
So, as I said, Erwin Friedovich discovered not only the first superoxide dismutase, but several others. And it was a real turning point in our understanding of essentially living in air. How do you live in air? Oxygen is a very, very oxidizing molecule, but it really hadn't been appreciated in biology how damaging O2 is. And it's really with the discovery that there was an enzyme that lowered the levels of superoxide. Superoxide is just O2 minus, one more electron, in vivo, that people realized, started really paying attention to antioxidant enzymes, and the the constant barrage of oxidative stress that we live with when we live in air. And so the whole area of oxidative stress and the oxidative stress-related illnesses and aging really developed around that time. And so although we were doing the inorganic side of it, we were always very aware of the relationship to oxidative stress and health.
Now, the term oxidative stress is another one that actually, it's another area that you do work on,
yes,
but obviously overlapped with the superoxide dismutase chemistry. Describe to us a little bit about oxidative stress and what that means chemically and maybe physiologically for us.
So that's actually been a big focus of our work, I guess because I came into the field really as a chemist, and people started talking in the field about the chemistry of reactive oxygen species, ROS. And people would talk about it as though ROS was a molecule. And ROS actually represented superoxide, hydrogen peroxide, hydroxyl radical, other related peroxy radicals, and so on. And of course, as a chemist, I was very aware that these have very distinctive chemistries. And so one of the things that we worked on quite early on superoxide chemistry was finding out what is the chemical reactivity of superoxide, how does it differ from hydrogen peroxide, and then what are the vulnerable targets in vivo. And then I ended up actually working with some in vivo systems in yeast where we made mutants that had no superoxide dismutase, and then we looked at what was wrong with them and to try and deduce what the problems were. And the really interesting thing that happened from that is that they had it was all about metals, problems with iron metabolism, differences with levels of manganese. So the whole issue of oxidative stress and superoxide has always kept me very metallocentric.
It's actually one of the things that I will, when I'm teaching even freshman chemistry, and we talk about molecular orbitals for O2 and the fact that molecular oxygen is a triplet.
Right.
And that keeps us all from burning up.
Right.
Yeah. And so it's very important.
Yes.
And then the species that form upon reduction, partial reduction, those are the ones that can get you.
Right, that's right, that's right. And they each have distinctive chemistries, and I think it's pretty interesting. It really wasn't appreciated back in the early days. And so it's been a very interesting field to be in over these years.
What would you describe as your, let's say, proudest research accomplishment and perhaps your most important one? It's sometimes they're different, sometimes they're the same.
I think, let's see, so the ones I was most excited about are not the ones I was necessarily the proudest of, but I can tell you all of them.
That's all right. Go ahead, continue on.
Okay, so the one I was most excited about was, when I realized that a coordination complex we had made by reacting an iron 2 porphyrin with superoxide, and it reacted 1-to-1 and made an adduct, but it had, I knew it was iron 3, but it had a rhombic EPR spectrum, and this made no sense because an iron 3 porphyrin should not have had a rhombic EPR spectrum. And this was really bothering me, and I actually remember I was at the Metropolitan Opera, when I realized that the coordinate, that it was binding sideways. And that was why it had a rhombic EPR spectrum. And I think that's the most exciting thing. The thing I'm most proud of probably is having, or actually proudest of my students for doing. They really decided they were going to make a whole large range of mutant superoxide dismutases, the different ones that cause ALS. And this was a huge amount of work on their part, not on my part, but on their part. But the result of this is that we had these different mutant SODs and we could study their properties, and we were able to group them into some distinctive classes where the properties differed, and that hadn't been realized before that time. In the ALS field, and so I think that's what I'm proudest of.
Now, you actually have bridged biological chemistry and inorganic chemistry, and have been a leader in bioinorganic chemistry for years. What advice would you have for your students and others and new faculty in terms of communicating their science, how to frame their science, and to whom to communicate their science?
How to communicate their science. How you communicate your science is a real challenge for a young person. I mean, I remember people telling me, people who were my mentors telling me, "Ooh, that's really exciting. You've got to publish a series of communications about that." And I had a really hard time doing that because I like to know the answer to a problem. On the other hand, I would tend to wait too long to publish stories. And so, I had to find some way, some compromise where I felt that every paper I published was significant, but I wasn't waiting forever to publish things. And also, I think publication, when you publish a series of papers on a topic, the topic is evolving, and you have to realize that you're going to understand it better in the later papers than you do in the beginning, and that's okay, because that's what scientific publication is. I mean, it's like an ongoing conversation with the scientific community. And so you want to be sure that your data are right, and you're interpreting to the highest level you can, and you need to talk to lots of people and get their feedback before you actually submit a paper so that you can sort of pre-referee it. And then just get your story out there and do it in good journals like Inorganic Chemistry.
We're not pushing for an endorsement yet, but we'll be back to that, yeah. But also you have these different audiences. You have the more biologically attuned scientists and the perhaps the coordination chemists and the physically oriented inorganic chemists. And you want to make both groups of people well aware of what it is, because they're the ones that can have the perspective on this kind of problem.
Right, right. And so I think people like me have had to be sure that we are publishing in a range of journals so that we are talking to the audiences that are interested, because, I mean, for me, since I got into this biomedical stuff, I really have, I mean, people in the ALS field are not going to read, unfortunately, inorganic chemistry. Although they may be starting to now, I don't. But in the old days. So you try and publish in a range of journals, and to stay and to make yourself part of the different communities. So I've always identified myself as an inorganic coordination chemist. But I'm comfortable now with the neurochemists and people in neuromedicine and biochemistry and so on. But you need to publish in a diverse range of journals and be sure that you explain your work to a wide range of people.
Can you also accomplish that in part by going to conferences, judiciously choosing which conferences, and actually starting to make connections across disciplinary boundaries, I'll put that in quotes, by doing that?
Yeah, no, that's very important. And it did feel very strange the first time I went to a very biological meeting that had no inorganic component to it. But you know, I felt sort of lonely. Where are my friends, my inorganic friends? But you know, you meet people, you talk to people, and the other thing is you go to those communities and people are really interested. You know, metals are so important in biology, and so many people who work in biology areas don't really have a deep understanding, but they know that the metals are important, and they have all kinds of questions, and you become an instant expert. It's quite gratifying, actually.
John, let's turn our attention to the funding situation, which is really important for all of us, because it's the only way we get our science done.
Yes.
Yeah. Tell us a little bit about the funding situation you encountered when you started, and how it compares with the funding situation today for young investigators.
Well, so when I started, I don't think I wasn't very sophisticated about. I just wrote a grant and sent it in. But in retrospect, I realized that I really had a much easier time than young people are having now, because I didn't have to resubmit a grant very often. And you know, first I got NSF funding, and then I got NIH funding, and then I got another NIH grant, and so it grew, and I sat on study section, and you know, everything seemed very fair. But I was actually on the NIH study section at a time when things got much harder, and then all of a sudden I saw young people who were submitting over and over and over again, and I felt very bad for them. But the fact is that the people who did the good science and were persistent, they did get funded, but it is different. Actually, at one point, the person who mentored me about applying for grants and how to choose a research topic and so on was actually Fred Hawthorne. So I went to Fred, before I submitted for my second NIH grant, I went to Fred, I said, I don't have enough money. I don't, you know, I have to apply for another NIH grant. Can you give me some advice? You know, they're never going to give me a second NIH grant. And he said to me that the one thing that the best advice he could give, and the thing that he followed himself, was you figure out what kind of science you really, really, really want to do, and then you apply for it. You describe it, you apply for it. And if it doesn't get funded, you don't change your area. You stick to the same area, and you say, well, I must not have explained it to them right. And you keep persisting. He said, if you can't persuade those people, then you look for other grant agencies. But remember to always work on the science that you really, really are excited about and want to do. And that actually has been incredibly powerful advice for me over the years, and advice I give to other people.
It is great advice. And the other is the question of continually, not necessarily repackaging, but basically to address those weaknesses that might have been pointed out and go back again.
Right, absolutely. And somehow or other, young people seem to think that the members of review panels are super wise, but you know, they're just like all of us, if you don't explain it clearly enough, they may not have gotten it. So don't think that, don't think that you were, there was something wrong with your idea. You just need to try and explain it better, think clearly, address whatever they said, and be persistent, politely persistent.
Okay. Over the last 3 decades, we've seen a tremendous increase in the number of women in science, number of faculty. We still have a very, very long way to go. Describe to us a little bit about your own situation from that standpoint, and what advice would you have today for young women going into science and going into academic science, and how to balance the demands of research life and a faculty life and their personal lives?
Well, it is very different. I'm so glad it's different now. And actually, I'm very proud to be at the UCLA Department of Chemistry and Biochemistry, where we have a large number of women. And I was actually the first tenured or tenure-track woman in the chemistry department there. I was also the first chemistry PhD from Princeton who was a woman. So it was pretty lonely back in the beginning days. And in inorganic chemistry, I went to an inorganic Gordon conference where I was one of 2 women. And they put us in the infirmary, so right, because they didn't have mixed gender dormitories. It's a lot better now, but it still depends a lot on where you go to school, where you get a job, and so on. And I think that the most important advice I can give is to young women, well, actually to young men also, is to be very cautious about where you want to work, who you want to work for when you're a graduate student or a postdoc, and where you want to work when you start to interview and get job offers. You want to be someplace where you like the people and feel comfortable and feel respected. And I think this, I can't emphasize this too much, even if it's, the place that you always dreamed of, when you visit, if you don't get the right feeling about it, be cautious. And then, and then go for the opportunities when they present themselves, and good luck.
How about the question of juggling all of your demands? We all are jugglers because we have many things to do, and in many ways I think women have it more, it's tougher, and I would like to try to have that ease so that we can have gender equal juggling. But in any case, what would you say to young women who also think about their own, the family, personal lives as well as their academic lives?
Actually, the demands on young couples these days, it's both the young men and the young women. If a couple decides to have, if they have a dual career and they have young children, it's a burden on both of them. I was asked by someone in my lab recently, when is the best time to have children? And I said, there is no best time to have children, but if that's what you want to do, you need to do it, and you just figure it out along the way. Some children are real easy to take care of, and some children are very demanding, and but you want to pay careful attention to how long your commute is and what kind of child care you have, and play it by ear. But it's very doable. There are lots of people now who've Oh, it's also important to have a supportive department, which UCLA has, and actually a growing number of departments are really giving, it's not just time off to tenure, but really being supportive of young men and women when their children are born, to be sure that they're not overburdened in those very hectic months and so on, which is again why I really urge people to think carefully about where you end up working.
Okay. How large a group do you have? And over the years, how many PhD students or postdocs have come through your lab?
You know, something I didn't, I was going to figure that out. I have a lot of PhD students. I've had fewer postdocs. I really enjoy working with graduate students. The postdocs who have worked with in my lab, actually some of them have ended up being senior research staff for a number of years before they went on to other jobs, and they've been wonderful. But I've really focused mostly on graduate students and also undergraduates, research undergraduates, which I've enjoyed very much. In terms of size of group, the group, I mean, at Rutgers, it started out small and it kept getting bigger and bigger. And when I moved to UCLA in 1980, a number of students actually came with me. And then I built up the group again. I mean, people added to that at UCLA. And now it's a little smaller than it was at the height, but 20 people, now it's smaller. Just whatever happens to be.
What advice would you have in terms of how to run a group, in terms of how we interact with all of our students and keep everybody moving forward in the most positive way?
So my advice is to really pay attention to what gives you yourself the most pleasure in terms of the level of involvement, of your own personal involvement in the research. So when my group has gotten large, we were still very, very productive, but I started running out of time to spend enough time with people, really going over data, and I'm really literally talking for my own personal pleasure. When I look at visible absorption spectra of some metal-containing species, I get a lot of pleasure from that. And if the group's too big, I don't. But I know other people who can get great pleasure and seem to be able to keep in touch with a whole lot of a very large group. So I think it depends on the individual. But one of the things that's very important is that each student or postdoc or undergraduate get the mentoring attention that they need, either from the advisor or from somebody else in the laboratory. And I think it's very much too bad when a group gets so big that some people get lost in it. And I don't think that's been the case in our lab ever.
Good. Let me turn attention to publishing. We've both been involved in publishing or publications. You are a very successful editor of a very successful journal. Accounts of Chemical Research has really set the standard in terms of an article type. The account, the perspective from a person's laboratory, and in terms of a specific subject, has one of the highest impact factors in chemistry. Everybody wants to write an account. tell me about the challenges and enjoyment you have in editing.
Well, I very much enjoy editing accounts. But I need to make clear that I did not invent the format of the account. That was Joe Bennett. And really, when Joe Bennett, the first editor of Accounts of Chemical Research, when he started the journal, this was a really totally new format. And so he was really a pioneer, and it's turned out to be very successful. I personally really enjoy editing it because I work with a small number of senior editors, and we read the papers. The volume of papers is not so high that we don't have. The volume of papers is not high. We get a chance to read them and to work with the authors in improving the accessibility for the non-specialist reader. And that was something that Joe Bennett started and has persisted.
Before you did your work as editor-in-chief of accounts, you were an associate editor for Inorganic Chemistry. I think many of our viewers don't know that or appreciate that. You did it for more than 5 years, part with Fred Hawthorne. Tell us a little bit about your experiences with inorganic chemistry at that time.
Yes, Fred Hawthorne was he's a colleague of mine at UCLA and really a mentor to me. And I was stunned when he asked me to be an associate editor of Inorganic Chemistry because I was so enormously flattered. I had no idea if I would be good at or would enjoy editorial work. But I was very flattered, and so I said yes, and then found that I really did enjoy it very much, and that working with Fred and Herb Case was very enjoyable, and the other associate editors. I was specifically asked to increase the profile of bioinorganic chemistry, and so that was an exciting time with bioinorganic really growing then. And Inorganic Chemistry as a journal, the major journal in inorganic chemistry, was paying serious attention to the growth of this exciting new area. So I really enjoyed it a lot, and would have stayed on doing it if I hadn't been asked to take accounts.
What are the particular challenges that you see for authors, researchers who write papers in areas like bioinorganic chemistry, and to whom should they address their papers, and where should they publish them, and why, from one standpoint, Inorganic Chemistry is an extraordinarily powerful journal to publish in, and where, from a different point, they may be looking to the people who read biochemistry or even medicinal chemistry journals. Yeah.
I think things have changed in recent years in a good way, because if you put the right words in your title and abstract, a much broader range of people will pick up your paper, even if they don't normally read in a specific field. So it's very, very important that you, if you're working in a biologically relevant area, but you have a very inorganic paper that you feel, that's your really desired audience, but you want the biological people to be aware of it also. With the proper words in the title and the abstract, often people, I at least, read papers based on very broad searches and keywords, and I read papers from over a huge range of journals, but just using those search services. And so I think things are getting better in that respect.
where would you find the keywords or the particular vocabulary to use for wider attention?
Well, in the beginning, when I was publishing on superoxide dismutase relevant to ALS, it was really a challenge because we were doing some inorganic spectroscopy with these mutant proteins and so on, and I mean, it really belonged in the inorganic literature, at least the chemical literature, and yet I felt it was really relevant to the biomedical people. So I would have to put, amyotrophic lateral sclerosis in the title or whatever, and then people were reading it. So you really have to you need to be sure that you think about the broader audience when you write your title and abstract.
How do you think communication of research results will change in the next decade or so? We will have journals, but they'll probably be paperless journals. And have you given thought to, actually I know you've given thought to how will scientific information be communicated in the future?
Well, I can't see the future with any certainty. I do hope very much that we retain the peer review system that we have. I mean, when I find in my searches a paper from Inorganic Chemistry, I know the high level of reviewing that it's been through. This is incredibly important to me. It's a validation. I hope we're not going to get to a place where we just all publish blogs. But that being said, I think the paperless world it's really exciting. I think that this ability to search across broad ranges and read over many, many different areas. I mean, the papers to read are brought to you by Google Scholar, Medline, Web of Science, etc. And I also think you can even browse that way. You just sort of keep changing keywords and see what pops up. So I think it's very exciting. I think it's getting easier to see what things are sort of related and that are not, to avoid having tunnel vision. I know other people say, oh, we're only going to read papers in our own specialty, but I don't find that's true of me at all. I find that I start reading all kinds of things because of my searches. Well, one of the things that's important is to remember successful search strategies, because you can, I mean, if I have a combination of keywords that give me really valuable papers in a field, I don't necessarily have to keep that search because I can reproduce it if I remember the search strategy. So yeah, it's a whole different way of thinking than we were brought up on.
That's right. Yeah. But I think you're more proficient than some of the other people that are, let's say, in our category, age-wise, with regard to reading the journals and searching and
well, computers are my hobby.
So that's
electronic gadgets are my hobby.
That's great. In any case, with that, let me thank Joan Valentine and also say to you that I hope you've enjoyed this interview and had a chance to really take in what one of our real leaders has to say about science, communication, publishing. Joan, it's been a real pleasure.
Thank you, Rich. I've enjoyed it.